So, Brian, do you want to give us an idea of what you're going to present while Venus here, and can at least enjoy some of this?
Yeah. Um, so how do I share my presentation?
You can just do a screen share.
So go up to the top. There's 3 dots, and then you. Yeah, there you go. You go, girlfriend.
Oh, you missed my snap.
Okay, guys, let's do the introduction. Hi, guys. Welcome to our journal club meeting today. Brian will be presenting over his awesome paper. Brian, take it away.
All right. So today, it's going to be sort of a non-traditional journal club because the way this was presented and published was sort of split up between 2 publications. So we have figures from the paper where they describe the actual algorithm, and also have figures from the companion paper where they describe the statistical methodology they utilized. So I guess to start, my first question is, out of the people here, how many of you are familiar with single cell analysis, specifically dimensionality reduction like t-SNE?
I've heard of it, but I haven't done anything with it.
I've played with it, but I haven't done anything formal with it.
You hear me?
Yeah, I can.
Hey, Alex.
Yeah, I know a little bit about it. I've actually went through, what's that dude from Harvard? Raphael Erzere.
Yeah.
He talks about it in his course for exploratory data analysis for high throughput sequencing.
Oh, nice.
Yeah.
Is that public anywhere?
So then, a sort of brief introduction on t-SNE. The idea behind t-SNE and most dimensionality reduction techniques is that for every cell, each dimension is a gene you've measured expression of. So we're not talking about dimensions in a physics sense, but just in a sense that there were some number of observations made about any given cell. So one of the most popular ways to reduce the complexity of the data is t-SNE. And this site is a great little site to go through in your spare time to sort of get an idea of what t-SNE looks like. So t-SNE itself just basically tries to take a group of data points in multidimensional space and map them down onto 2 dimensions. So what these little things are showing is that you start with your population data originally, and then as t-SNE cells get repositioned into the 2-dimensional space, and eventually they should converge at a solution. Is everyone following so far?
Yeah.
Like
PCA?
Yes, it's very similar to PCA. The thing to keep in mind is PCA is considered a true sort of transformation, whereas t-SNE is considered a mapping or projection.
Okay.
The other important thing to keep in mind is that t-SNE is stochastic. Every time you run it, it'll end up slightly differently, whereas PCA, it's consistent from run to run. The other thing to keep in mind is t-SNE has 2 hyperparameters. The first is perplexity. And the easiest way to think about perplexity is it's how much do you value global versus local variation. So if this is the original data and you have perplexity 2, you can see what happens. And as you change the perplexity, the sort of distribution of data points changes as you alter that. And you can see sort of you can go too far in terms of altering the perplexity. The other thing is step number, which is just the number of iterations. And again, you can sort of find the wrong number of steps to take. Like this is clearly way too overfitted for the data. And you would want to avoid that. But you can see sort of at step 10, you have good representation of the original data, but you also have good representation around step 120 or step 1000. So those are the sort of key things about t-SNE. Now, the reason I mentioned that is because Monocle is a trajectory mapping tool uses Tisney as a means to sort of start the process of modeling the data. So what Monocle does is you start with your multi-dimensional data set. So in this case, each point is a single cell for which you have some level of gene expression values. In the first step, you do a dimensionality reduction, usually using PCA, but they do support LLEDM. They also support UMAP now, which is becoming a little more popular in the single cell community. UMAP is similar to Tisney, but it is not stochastic. So if you run UMAP and I run UMAP on the same data set, we're going to get the same exact projection. So after they do the initial dimensionality reduction, they then find K clusters and they do this iterative process where the cells are slowly shifted towards the clusters it found, and then the clusters are refound and the cells are shifted again and again until ultimately you end up having sort of coerced all the cells onto this actual trajectory path. And then finally, as the user, you have to select what your parent population is. And in this case, you just look at expression of genes of interest, and you'll be able to see, oh, here's my stem markers. So this is the root of my population. And then Monocle will go in and then calculate pseudotime, which is just geodesic distance to a root cell. And then lastly, you can look at the branches that determine cell fate for differential expression analysis. So is the basic step-throughs of Monocle clear?
Yeah.
Mark, you still with us, or did you go get banana bread?
No, I'm still with you guys. I think so.
All right. I mean, please interrupt. Please stop me anytime. So over here, they're showing what the actual data analysis looks like. So in panel B, they have an ICA analysis of the cells, and then in panel C, they've applied their actual projection using what they call a DDR tree. And then panels D and E, they're just showing the accuracy and robustness of their ability to look at pseudotime using various trajectory analysis tools. Monocle 2, of course, is the tool they're presenting here. Monocle 1 was a previous iteration. That just used a minimum spanning tree instead of this DDR tree approach. And DPT and Wishbone, I forget what their underlying methods are. But if I remember correctly, they are sort of variations on probabilistic state modeling and minimal spanning trees. All right.
Okay.
No. It's hard doing a virtual journal club because I can't like see you guys and see your eyes glaze over.
Oh yeah, you guys, you're doing full screen?
Yeah, is that?
Oh no, it's basically if you want to see us, you can just exit full screen, for example.
You're telling me all these wonderful things that I've never realized before.
Oh, not go. Actually, going into full screen might help. No, but basically, if you're doing full screen on the presentation, it will lock out us, right?
Right.
But you can just screen share and keep us hanging below or something like that. It's whatever floats your boat. That's what I have set up, but
I'll learn this eventually. So in figure 2, and this is the last figure from that paper, I had forgotten that these papers are actually a lot more sort of formula-based and math-based than figure-based. So they're probably not the best papers to do for a journal club. But in this case, they've taken a single-cell data set and applied Monocle and then looked at how various knockouts of cells alter the trajectories. And so here they're looking at hematopoiesis, how this hematopoietic stem cells sort of differentiate into each of the multipotent progenitor cells. And then here they're looking at the same sort of trajectories, but now they've done various knockouts to show how you can sort of shuttle particular cell subpopulations into lineages by sort of blocking their ability to differentiate, focusing on these, I forget whether these are master switch regulators or what, but basically if you don't have these, you can't differentiate properly. Yeah. So knock out IRF8, everything, all the cells differentiate down to this cell type, whereas if you knock out GFI1, they tend to differentiate down to this cell type, and then if you knock out both, they're sort of unable to switch into each progenitor. I mean, this is just sort of the wet lab follow-up to their approach.
Yeah. So I have a silly question. This
Sure.
might be actually really silly. This is done on eukaryotic cells, right? Basically, multicellular organisms. So sometimes with bacteria, you can generate different phenotypes based on conditions and knockouts. Do you think this could be expanded into, I guess, bacterial populations as well, or?
So I don't see any reason why you wouldn't be able to do trajectory analysis of bacteria. I imagine that it would be very useful, especially in cases where you're running into pathogenic bacteria.
Yeah.
I think your big problem would be that how you do your single cell sequencing. I'm not sure if any of the kits are sort of targeted towards bacteria or archaea for that matter. It's been a while since I did microbiology, but I know, for example, the 10x single cell kits are all designed to grab onto poly A tails.
Okay.
Bacteria don't have poly A tails on their transcripts, do they?
For mRNA they do.
Okay.
But yeah, okay.
Yeah, I don't know how, if the kits would work, but if you have single cell data from a bacteria, it should work.
You can get RNA-seq from like E. coli and stuff like that. I know I'm going to work with RNA-seq data soon for that actually. So that's why I'm asking. It's just curiosity.
Yeah, I don't see why it wouldn't work as long as you have the single cell RNA-seq data.
Cool.
And I will say, from sort of an implementation approach, implementation is really well put together.
Okay.
If you go to their GitHub, it's really well laid out, especially compared to Monocle 1. And Monocle 3 is now in alpha testing, and they've been really good about keeping that sort of fork of their library really active and developing. So I looked at some other trajectory analysis programs before I settled on Monocle 2, and while they might have been a little more robust and they had slightly different methodologies underlying them, Monocle 2 at the time, and this was back in May, was definitely the sort of most put together at the time. The other ones I looked at was ERD and Cell Router, and both of them seemed like they were sort of a little more in the weeds. So if you like playing with other people's packages, check them out.
Okay.
But if you just sort of and shoot sort of approach, Monocle 2 fits that niche quite nicely.
Awesome. Thank you.
All right. So this is from the other paper, their census counts paper. And here I'm just going to focus on the aspects of that paper where they get into their differential expression analysis. So as I'm sure you guys are aware, when we do a differential expression analysis of RNA-seq data, we use generalized linear models. And we use them with a negative binomial distribution because that fairly closely represents transcript abundance in a cell. Now, there are a few flaws using that. when we say we're fitting the negative binomial distribution, we're actually a specific case of it. So it's not a true negative binomial distribution. Now, the other little hiccup is that in single cell data, we have a lot of dropouts. This is also known as zero inflation. If you ever want to go down an interesting rabbit hole, just look up papers dealing with zero inflation in single cell data, and you'll see there's dozens of new differential expression algorithms out there that explicitly address this. Monocle, too, chose to go towards what's called a vector generalized linear model. And the way to think of that is basically it's a generalized linear model that makes fewer assumptions about the negative binomial distribution. So from the get-go, it can handle lots of zeros. It can handle drop. It doesn't require your data doesn't require as many assumptions. It's basically edge R on steroids, and the runtime of the algorithm definitely reflects that. It takes like maybe 10 times as long to run Monocle's differential expression analysis than it would to run an equivalent edge R analysis. So over here, they're just showing the trajectories they've built out that they want to explore. And so here they have their sort of PCA looking at the expression of these different markers to sort of identify what the root of their population is. And then they go into ordering the cells, right? So that's that iterative process where you sort of find your K center clusters and you shift the cells towards those K centers over time until the true trajectory. And in this case, they've identified 3 cell states: your progenitors, your branch A, and your branch B. So what they now do is they want to look for to detect the branching genes. So sort of genes that change little in the progenitor part of the branch, but then drastically shift when you go either into branch A or branch B. So when we do this sort of thing in a regular differential expression analysis, what we would do, because this is essentially a time course, right? You have time 0, time 100. We would fit a spline. So have you guys run spline regressions in EdgeR or DESeq or Lemma before?
Nope.
No.
Okay. So if you have a continuous variable that you want to do differential expression of, you can use a spline. And generally it's recommended to have your degrees of freedom be between 3 and 5. It's sort of like taking your continuous data variable and binning it into different bins. So rather than trying to look for a linear relationship between your continuous variable and your gene expression values, you sort of bin it into however many degrees of freedom. And then you look at each bin in the spline. If you look at the EdgeR or Lemma manual, they both have a nice little section explaining this more in detail. I don't fully understand it myself, so I'm sure someone's going to watch this later and be like, that's not what a spline is at all. But that's how I like to think about it. Does that make sense?
Yep.
Yeah.
Okay. So what this now does essentially is it'll say, it'll fit a model for branch 1, for progenitor through branch A, and then progenitor through branch B, and it'll just ask, is the model for branch A different from the model for branch B? And that's what this is basically showing here. You know, here's the model for AT2, here's the model for AT1. That's different, so it has a significant p-value. Contrast that with these two, where you can see the the model for branch A and branch B, or AT1 and AT2, looks almost identical in these two cases. So that's all it's doing. And you can see like the sort of curves in the line. That's the sort of effect of the cubic spline. It's not a linear relationship, but it's this cubic curve that gets fitted. So when you apply this sort of whole scale to a trajectory analysis, you end up with something like this heat map. Where each row is a gene, and then you have your trajectory 1, your trajectory 2, and you can see how the expression of these genes change as you diverge further from the progenitor into the cell fates you're concerned with. So you can see over here all these, in this case they're modeling GO terms, but these were derived from their gene expression profiles. Here you can see over time they have upregulation in your normal cells, whereas in the knockout, these cells, actually, the expression of these GO terms really drop off. And you can sort of scroll down the heat map and see the other sort of responses they're quantifying. So that's sort of monocle in a nutshell. And those are sort of all the figures from those two papers that really apply to that particular aspect. So were there any questions? Was the banana bread good?
Banana bread is out in 6 minutes.
I thought it was 7 minutes, like 10 minutes ago.
No, it was 37 minutes, like 37 minutes ago.
Oh.
Or 31 minutes ago.
Yeah, so as I said, the number of figures in these papers is sort of really short, and so I didn't even realize that.
But they're pretty good figures, to be honest.
Huh?
They're pretty good figures, to be honest.
Yeah.
Like, they communicate a lot, especially with the information you're providing.
Yeah, and so I tried to cover as much of the math in as an approachable way as possible.
Yeah.
So please hit me up with questions. I know it's also a lot to deal with because, we're going from, we have to cover single cell, dimensionality reduction, trajectory building, and then also this niche differential expression analysis tool.
I mean,
is this part of is this package going to be part of the other paper? Single